everyone. Today we are going to learn to study the topic generation and conduction of nerve impulse. Now all of you know that what do you mean by nerve? The nerves are the structural and functional unit of nervous system, and the function of the nerve is to carry the message from brain and spinal cord to different parts of the body and from different parts of the body to the brain. So the function of the nerve is to pass the messages in the form of impulses. This is the structure of a neuron, typical neuron. This is multipolar neuron. Okay, let us revise quickly. This structure is called as cyton. Centrally placed nucleus means this structure is cyton. This structure is nucleus. These structures are dendrites. This elongated structure is axon, and these are the axon terminal. Okay. Now this membrane of the axon is called as axolemma, and the cytoplasm of axon is called as exoplasm. This is the membrane as it is present around axon, axolemma, and cytoplasm which is present inside the axon called as exoplasm. I think till this it is clear. This is the basic structure. Now the thing is that when the nerve is carrying impulse or the message, the nerve, it consists of or it goes in the three steps or the three stages. The first stage, it is polarized state. It is also called as resting state. Second, depolarized state, and last is repolarized state. Polarized state, it is also called as resting state. So first we will concentrate on the polarized state. Polarized state, as it is also called as resting state. Why it is called as resting state or resting stage? Because the nerve is not carrying any impulse or any message. Okay, that stage or that step is called as polarized state. Now here we know that the meaning of pole means the charges which are present on the membrane, positive charge and the negative charge. Okay, let us come to this diagram one more time. This is axon. The same diagram I have here focused and enlarged. Here I have drawn the membrane. Axolemma I have drawn. Okay, this particular part I have drawn here. Okay, so details I have shown. Which structures are present in this axolemma I have shown. Okay, before that, just come to this diagram. If we study the outer fluid means outside the axon is called as extracellular fluid. The fluid which is present inside this exoplasm is called as intracellular fluid. Okay, extracellular fluid means the fluid which is present outside the axon, the fluid which is present inside the axon. ECF, extracellular fluid, ICF, intracellular fluid. Now here, if you observe, you will find that outside this axon, more sodium ions are present as compared to inside. In case of ICF, that is intracellular fluid, more potassiums are present as compared to outer fluid. It means potassium ions are present outside. But if you see the ratio, more potassiums are present inside. If the sodiums, they are present more in number outside, but they are present inside also. But comparatively, more sodium outside and more potassium inside. Okay. Here, chloride ions are also present. Inside this, the negatively charged proteins and the nucleic acids, they are also present. So if we have to remember, you can remember the salty banana. In case of salty banana, potassium, the banana consists of lot of calcium. So lot of potassium. So this potassium, it is present inside, and as I said, salty means sodium chloride, it is present outside. So here I have drawn more sodium and inside I have drawn more potassium. Now the thing is that just come to the charges which are present outside the exolemma and inside the exolemma. Okay, here the outside, when the nerve is not carrying any impulse, at that time this outer surface is positively charged and the inner surface is negatively charged. The reason behind is this: more negatively charged protein molecules are present inside, though potassium is present. Here sodium ions are more. So this polarity, it is maintained. And if we measure this polarity with the help of voltmeter, you will find that when the nerve is not carrying any message, at that time the resting membrane potential, it comes minus 70 millivolt. This is the number if you measure it. Now how to maintain this polarized state? For that, here the three things, they are continuously operating on these membrane. The first thing, sodium and potassium leakage channels are present on this membrane. Second thing, voltage gated channels are present on this membrane. And the third thing, sodium potassium pumps are present on these membrane. Now let us see. Here with red color I have shown potassium leakage channel. With green color I have shown sodium leakage channel. Now what do you mean by leakage channel? As the name itself indicates that these ions, they continuously move in and out. Now this membrane is more permeable to potassium as compared to sodium. In other words, you can say that more channels are there for potassium and few channels are there for sodium. So continuously the sodium and potassium, they are coming in, going out, coming in, going out. That is the main function, as they are the gate, or you can say they are leakage channels. So continuously they are getting diffused from higher concentration to lower concentration. Now come to this pump. This pump is called as sodium potassium pump. Now as the name itself indicates that they have to pump it. What exactly this pump is doing? 3 molecules of sodium. As sodium is present outside more, it get easily diffused from higher concentration to lower concentration because sodium is more here. Okay. Potassium from higher concentration to lower concentration, it will move out. But the function of this pump is it just maintain the original position of this membrane. Means as soon as the sodium ions, they leak out and potassium, they leak from this side, continuously this structure, it rotates. This pump, it rotates. And whatever sodium ions, if they are entering, continuously 3 molecules of sodium, it moves out, or pump forcefully, it just forces out, and potassium, whatever they have entered here, with the help of this pump, it just, what you can say, get back into the exoplasm. So continuously this process is going on. 3 molecules of sodium coming back to the outer surface and 2 molecules of potassium, it is coming in. Due to this, though here the sodium, it is coming inside, potassium is going outside, with the help of this pump, this ionic equilibrium is maintained. Now the third thing is the voltage gated channel. There is voltage gated channel for sodium. There is a voltage gated channel for potassium also. What is the function of this channel? When the nerve is not carrying any impulse, means polarized state or resting period, these voltage gated channels, they are closed. Only these two things, they are operating. Now the thing is that this pump has to force to get back these ions to their normal position, and for that it requires ATP because it has to work against concentration gradient. But when the nerve is not carrying any message, these voltage gated channels, they are closed. But at what time they will open? Whenever there is a difference in the potential, automatically these two gates, they are going to open. We are studying generation and conduction of nerve impulse. We have already studied that there are three stages. The first is polarized stage, second is depolarized stage, and third is repolarized stage. The first one, polarized stage, when the neuron is not carrying any impulse or any message. That neuron is called as polarized stage, means that neuron is in polarized state, or you can say that it is in the resting stage when not carrying any message. At that time, now this is axolemma, the covering plasma membrane of the axon. Outer side is positively charged due to presence of sodium ions more. Inside means exoplasm negatively charged due to presence of more negatively charged protein ions. Here at that time, many or most of the leakage channels for sodium and potassium, they are closed. Very few channels, leakage channels, they are open. And if we measure the potential during resting stage, it comes minus 70 millivolt. So this is about polarized state or resting stage. When the neuron, it convert or it goes in depolarized stage. Now when it will go in depolarized stage, as soon as the nerve is getting any impulse, at that time the sodium channels, they open. Here with red color I have shown sodium channels. So as the sodium channels open, here more sodiums are present. Outside means extracellular fluid. This is intracellular fluid. Here more sodium ions are present. They start coming inside the exoplasm. And here this what you can say, the entry of sodium, it takes place along with concentration gradient because sodiums are more outside and here potassiums are more. But here these potassium channels are closed. So there is no exit of potassium. So naturally here in case of depolarized state, the positivity of the inner side or the inner part, it increases, electropositivity increases as compared to outside, and hence the polarity, it changes from positive to negative and from negative to positive due to influx of sodium ion, and the stage is called as depolarized stage. Now if we measure the potential, as it is going towards the positivity, it changes potential from minus 70 millivolt to plus side, plus 30 to plus 40 millivolt. Let us study about repolarized stage. Now in case of depolarized stage, there is the influx of sodium. More sodium channels are open. Here potassium channels are closed. So more and more sodium from outside, it enter inside the exoplasm. So due to which the inner side becomes more and more electropositive and the outer side, it becomes electronegative, and the polarity, it changes from minus 70 millivolt to plus 30 to plus 40 millivolt. After this, the neuron, it enters into the third stage, and the third stage is called as repolarized stage. It means after depolarization, again it enter in polarized state. So it is called as repolarized stage. At that time, now this is the axolemma. Here the sodium channels, they are closed, but potassium channels, they open. As potassium channels open, more and more potassium, it just run out of this what you can say axon, means it enters in the extracellular fluid. As more and more positivity, it moves outside the inner layer or the inner membrane, it becomes electronegative, and the outer membrane, it becomes electropositive. It means now the potential, it again restored to minus 70 millivolt. But sometime what happened, these leakage channels, potassium leakage channels, they remain open for a long time. So more and more potassium it goes out, due to which more and more negative charge is restored, and that potential it may goes to minus 90 millivolt also, which is called as hyper repolarized stage. Let us study how the impulse runs all along the fiber. Let us study. Now we have studied there are three stages. The first stage is polarized state, means resting stage. Then it enters into depolarization, and last it enters into repolarization. But how the impulse it runs along with this fiber, let us study. We know that when the neuron is not carrying any message, the stage is called as polarized state or the resting stage. At that time, the inner membrane is negatively charged and outer membrane is positively charged, and the neuron is said to be polarized. Positive charge outside and negative charge inside. Now as soon as these neuron gets the impulse, this particular area or that particular zone, it becomes depolarized by rapid influx of sodium ions. These sodium ions, they enter inside the axon, so that now this charge it becomes positive and this charge it becomes negative. This condition now it is called as depolarization. Depolarization means rapid influx of sodium inside the exoplasm. Okay. Now, if you see here positive charge is there, here negative charge is there. These two poles, they get attracted towards each other, or you can say the electricity it passes from positive pole to negative pole. Okay. So, as the electricity pass here, this particular zone, again it becomes depolarized. Rapid influx of sodium takes place. So here it becomes positive and here it becomes negative. Again there is a electricity, current passes from positive to negative. Okay. So again this particular membrane or this zone become depolarized. So again it becomes positive and here it becomes negative. And in this manner the current it pass on along with the fiber, that the axonal fiber. Now if you see here, current always it passes from positive to negative. Two electrodes are there, so current always passes from positive to negative. This is one thing. Here the current, here positive charge and here negative charge is there, so current can flow from this to this direction as well as from this to this direction, because here positive charge is there, here negative charge is there. Again current it can flow in this direction, current can flow in this direction also. Again current can flow in this direction and current it can flow in this direction. It means along with the fiber the current is bidirectional, but when it comes to end, axonal end, okay, means in the synapse, at that time it becomes unidirectional in case of chemical synapse. This is all about the impulse, how the impulse run along with the fiber. Now if we study in case of medulated nerve fiber, we have nodes of Ranvier. So these impulses, they jump from one node of Ranvier to another node of Ranvier, from second node of Ranvier to third node of Ranvier. Such type of conduction is called as saltatory conduction in case of modulated nerve fiber.